All right. Again, we are incredibly happy to have you all joining us as we kick off the Fall 2023 Pet Food Alliance webinar series. For many of you, you've been long-term members of the Pet Food Alliance, but for some, this may be your first interaction with the Alliance. And it's important for us to remember what the Alliance was founded upon and remains committed to, and that's the collaborative development of solutions to industry-wide challenges and the exploration of mutually beneficial opportunities in collaboration with pet food industry members, rendering industry members, allied industry members, and academia. We're excited to kick off our educational series as the Pet Food Alliance moves towards educational opportunities to inform not only members of our community, but future members of our community. And joining us today, we have Dr. Valentina Trinetta from Kansas State University, where she is an associate professor with a world-renowned reputation in food safety, and will share with us some of the research happening at Kansas State University that stemmed from a conversation in our product safety working group. We'll continue our webinar series later this fall with webinars focused on advances in peroxide values, as well as innovation in rendering and pet food manufacturing. I would also like to do a brief reminder for our upcoming working group meetings in Auburn, Alabama. These meetings will be hosted on November 15th and 16th, following the Imaginarium hosted at Auburn University with Balchem. We're excited to convene our working groups to continue our conversations about important research priorities impacting the pet food and rendering industries, and hope that you can join us. More information about the working groups can be found by emailing the PFA email address or my personal email address, or by phone. And we'll be sharing information about our final agenda, as well as some of the opportunities for networking interaction outside of the formal meeting itself. But with that finished, I'm going to turn it over to our speaker today, Dr. Trinetta, to share with us some of the exciting research that's happening at Kansas State impacting the pet food and rendering industries. So Dr. Trinetta, if you wouldn't mind sharing your screen, we'll get started.
Okay, so first of all, thank you for the opportunity. Today, my seminar will be on the influence of cleaning and sanitizing on the microbial ecology of trucks and tanks in the rendering, transportation, and pet food industry. From the introduction to this seminar, you understood that this project came about after a conversation within a working group. So I would really like to thank that working group, because this project has been fun and very interesting. So if we look at the last 10 years of pet food recall, you can see that the majority of recall are either linked to kibble or can. And if I'm going to look at which biological hazard were linked to this recall, you can just see in the last 3 years, so 2022 and 2021, that the majority of recall are linked to Salmonella. And doesn't matter if it was dry, cooked, refrigerated. Salmonella and sometimes Listeria are foodborne pathogens that are linked to this outbreak. We start hypothesizing of what were the possible causes that were linked to the Salmonella outbreak in the pet food industry. And cross-contamination of the final render product was identified as a potential food safety issues. We also think that handling, moisture, temperature condition introduced after the rendering process, so during storage and transportation, might lead to Salmonella present and growth. And so we started this project, and I can share several peer-reviewed articles where we verify that introducing moisture and depending on the different temperature condition Salmonella was able to survive over time in poultry fat. We also know that the rendering industry worked very hard to try to get all the raw material and delivering to the pet food industry in a safe manner. That's why they have such a high thermal killing step. Based on all these observations, we kind of went towards the route of, yes, transportation and storage is probably the contamination point in this supply chain. So little information is known regarding how tank and trucks are clean and sanitized between transportation of the rendering fat. And after confirming that Salmonella can grow in hard condition and it is influenced by moisture, we decided to understand what were the conditions and to minimize the risk of microbial growth and recontamination on these trunk and trucks. So the first objective was to communicate with stakeholders and understand the current practices of cleaning and sanitation in truck washing facility. And I couldn't do this objective without the Pet Food Alliance, the Pet Food Institute, and the Fat and Protein Research Foundation that offer me really several occasions to talk with the working group and with several stakeholders. We were able, therefore, to obtain a SOP. And this is not universally used, but we think is a good example of what in this facility they do for cleaning and sanitize tank and trucks. So first of all, they drain the previous product. They remove all the gasket that needs to be washed and sanitized in a separate way. They start the cleaning place with the degreaser, with the sanitizer, and then the part of the SOP that was not consistent among the facility that we visit was following the sanitizer with a hot water rinse for a certain amount of time. But all the facility that we visit use sanitizer. At the end of this the CIP cleaning in place, the operator will visually inspect the trailer for cleanliness, usually using a flashlight, will allow for drying, and then will apply seal to opening, and they will be ready for the next trip. The objective too, after we identify this SOP, was to evaluate some environmental indicator and enumerate them before and after the treatment and while the operator was washing and sanitizing. So the experimental design included 3 facilities located in the Midwest. We tried to sample 6 trucks per facility. We identified 4 sample locations per truck before and after wash. We enumerate E. coli, coliform, and Enterobacteriaceae before and after wash. And we choose this indicator because they're usually giving an overview of the hygiene of the facility and the operation. And then when possible, we extracted genomic DNA, and we did an amplicon sequencing analysis for metagenomic. Our purpose was to understand which type of taxonomy we were having during this process. We, as mentioned before, we identify 4 sampling location: the manhole, the upper outlet, the pump, and the outlet. And this is kind of a schematic of the truck that we were working with. And in case you are not familiar, before starting this project, I was not. This is the manhole, so the sampling was happening using a lift to allow the operator to get the swab. This is the upper outlet. That's before cleaning. This is the pump and the outlet. And this is Colton at one of the washing facility, trying to get his sample while these people were working. So we also appreciate the flexibility of the washing facility in having us there for a couple of days while they do their job. And so this is a video of a truck entering the facility, and so ready for Colton to get his sample. And this is a video of while they are washing and sanitizing the truck. Okay, a little bit more specific of what we did from the microbial point of view. So we use environmental swab, so just sponges. And after we got our sample, we transfer the sponges in buffer peptone water. We perform serial dilution, and we use Petri film to enumerate indicators such as coliform and Enterobacteriaceae. When we got a significant amount of bacteria in our Petri film, we assume that we had enough DNA to do DNA extraction. And so we extracted DNA from our buffer peptone water, and then we did amplicon generation and sequencing, some bioinformatic analysis that is still ongoing. And I do have some data visualization to make you understand which type of correlation between sampling site track we have and which type of microbial environment we observed. Some results. Okay, the first couple of tables that I'm gonna show you are the microbial enumeration. And I should tell you that the facility where they do cleaning, degreasing, and sanitizing, followed by 10-minute hot water rinse, was very clean both before and after wash. So all the truck in entrance, before starting any type of cleaning and sanitizing SOP, did not have any recoverable E. coli or Enterobacteriaceae. Therefore, neither after their post-wash we were able to detect bacteria. Facility B did cleaning, degreasing, sanitizing, but did not follow their sanitizer step with a hot rinse water. Before washing, we were not able to see any E. coli or coliform, but we did see Enterobacteriaceae in the pump and the outlet. And very interesting, after the wash, we still observe the same for E. coli, but we see the reverse for the pump and outlet that are clean, while the manhole and the upper outlet that were below detectable limit before show a slight increase on Enterobacteriaceae. The third facility did not have a pump. They did cleaning, they did degreasing, they did sanitizer, and again, they did not follow with a hot rinse water for 10 minutes. These data to me make a little bit more sense, where I was not able to see any E. coli and Enterobacteriaceae in the manhole and the upper outlet. We saw both E. coli and Enterobacteriaceae present in the outlet before washing, and we have a decrease of this count after washing. Some partial conclusion is that washing step and location were both significant factor for the count of indicator. Pump represent the location where we found the highest count of Enterobacteriaceae. The relation and interaction between washing step and location were also significant for Enterobacteriaceae, but no location or washing step were significant for coliform count. We do think that implementing a 10 minutes hot water rinse after cleaning and sanitation might make a difference, because in the only facility where they did this practice, we observe a very low count of bacteria before and after washing. And we also think that developing an SOP that specify for how long the truck need to stay at the washing facility, so the lead time of the truck at the facility, might help to solve the difference in these cleaning and sanitation results that we got. What we think that based on how busy this facility were, some trucks stay there longer than other, and therefore allowing the possibility of biofilm forming. And we know from literature that it's very difficult to remove and control biofilm once they are formed. So none of these sanitizer practices will work if a biofilm is formed on these surfaces. I want to say that the metagenomic analysis is still ongoing. And we are still collecting some data. So this analysis is just a preview of some of the results that we will be then be able to share. But I wanted to show you, to tell you that because of the indicator counts, we were only able to extract DNA on 3 sample. And these 3 samples belong to 2 trucks. Pump 1, pump 2, and outlet 1. This is a graph that shows a relative abundance of taxa of each sample that was collected. And you can see that the majority of bacteria that were found on the pumps and on the outlet belong to Pseudomonas and Acinetobacter. This is another graph representing more or less the same information, but this time we graphically represent it through a heat map to show with red where there were which bacteria and where they were highly abundant as compared with the light blue where they were not very much abundant. This pump number 1 belonged to truck A, while pump 2 and outlet 1 belong to truck B. And again, you can see that Pseudomonas is highly present, as well as Acinetobacter in truck number 1, but then this graph allows us also to divide better the taxonomic abundance in a cluster map. And so we were able to see that also Clostridium is present in pump number 2. And in pump number 1, we found some bacillus. So even if we didn't go through serotyping, the presence of, for example, Bacillus and Clostridium is telling me that some anaerobic contamination is happening, and Bacillus and Pseudomonas are very good biofilm former. So thinking about the results that we got for facility B, where we observe that we do have some dirty spot pre-washing, that they get clean after washing, and the other 2 sites instead seems to get cross-contaminated, I think is the game that these bacteria are playing in moving around and finding new spot to hide and replicate. We were also able to do a principal coordinate analysis, and this analysis is trying to relate if sampling spot has any correlation from the microbial point of view. And as you can see, all these points are very far away, so that means that they are very different. And our analysis was not able to find any overlap between sampling sites. This is telling me that different facility, different truck, depending on what they transport and where they are located, might have different type of risk. So we cannot say, for example, that truck A was similarly contaminated as truck B. That's the meaning of this graph. Objective 3 was the development of an SOP that will help improve cleaning and sanitation. And as you remember, the initial SOP that I show you, the last part was highlighting the fact that operator perform a visual inspection. And so what we wanted to do is to give to the industry a tool to understand if we could standardize visual inspection of tank and trucks. So what we did is we use LED and UV flashlight, and we took picture pre and post wash, and of course before swabbing, in order to understand if this picture could give us a good visualization of what was clean and dirty, and we try to correlate this picture to our counts. This is the picture that we collected for the upper outlet pre and post wash. This is a LED dirty, and this is a picture of the outlet clean. So I'm not sure we observe a lot of difference, but a better visualization is obtained when I use UV. So this is UV dirty and this is UV clean. We did the same for the pump. So this is when we use LED, and this is when we LED dirty, so pre-washing, and this is LED post-washing. And you can see that the reflection is more or less similar. And this is instead UV dirty and UV clean. And then we did the same for the outlet, pre and post wash with LED and with UV. And finally, for the manhole, that I would say is the most difficult to clean and sanitize. LED and UV. We did some statistical analysis, and we concluded that using LED, we were able to correctly identify only 40% of the sample as clean or dirty, while UV flashlight correctly identify 66% of the sample as clean or dirty. UV was able to effectively identify and fluoresce residual poultry fat. However, we weren't able to correlate the presence of the fat residues with the count on Enterobacteriaceae that we obtained. And we concluded that based on our statistical analysis, we were not able to correlate microbial indicator with the use of a visual inspection by LED or UV. Nevertheless, I would say that the use of UV light might help to understand if a surface is qualitative clean or not, but not from the quantitative point of view. As I told you, the metagenomic analysis is still ongoing. So these are some partial results of our project. Again, I want to thank the Pet Food Alliance, the Pet Food Institute, the Fat and Protein Research Foundation, and all of you that stick with me during this seminar. So now, if you have any question, any suggestion, we are still ongoing, so any feedback might be useful to improve the research. So thank you so much.
Thank you, Dr. Trinetta, not only for this great research and informative research that your group is performing, but being willing to share your progress as well as some of the preliminary results today. Feel free for those of you who are joining us to either, if you have questions, to either enter them into the chat or unmute yourselves so that you can ask questions of Dr. Trinetta. I'll start first maybe with one that's come to me, Dr. Trinetta. When you finish this study, what do you hope are the outcomes that will be used by either the rendering and or pet food industries? What is the ultimate goal as far as implementation of the outcomes?
So I think these results show that the hot water rinse after sanitizing might be useful. And so the overall goal, that also in collaboration with the Pet Food Institute, was to finalize and optimize an SOP to share with the stakeholder and ask them to try to be as compliant as possible. We also think that setting some expectation of how long a truck needs to stay in that washing facility in order to be clean, and working with the operator in the washing facility should be addressed, because again, I think we observe a lot of difference between facility because of their different SOP, and so saying we expect maximum 2 days, or we expect 24 hours as a lead time, but no more than 4, will help also to decrease the possibility to forming biofilm. I do think again that those type of bacteria that came out in the taxonomic analysis are very good biofilm former, and the fact that in one of the facility we saw that some spots were dirty and they get clean after washing, but after washing other spots get contaminated, is telling me that cleaning and sanitize but not flushing might help just moving things around and not getting them out. So these are kind of 2 points that we would like to make. We were a little bit disappointed on the fact that our visual analysis didn't work out, and we tried both the 2 type of light, but we try also gloves. But I think it's depending on the type of metrics that we are working with being fed, and so maybe a little bit difficult.
Yeah, thank you for that. And this is certainly something from an alliance perspective, the creation of an SOP or some standards around sanitation for transporters is such an important priority, and it's exciting to see this work kind of lead towards that. A question in the chat, would you suggest to proceed with challenge tests, i.e. inoculating a surrogate organism into the tanker and enumerate pre and post for validating the sanitation method? Do you think that's next for your group?
I hope to be able to convince the Fat and Protein Research Foundation. I appreciate the suggestion. I think, yes, a validation study is needed. And will be useful also to touch base on those 2 points of leading time, and it is really useful, the hot rinse water. So yeah, thank you so much for your question. Yes, we do need that.
Exciting times. I think there's a lot of opportunity for research in this space. And I just want to go back to when these conversations first started from a product safety working group. The transportation sector really wasn't at the table, and there was a lot of learning to be done for members of the rendering and pet food industry about what happens during transportation. So I'm just curious, from your perspective as a research scientist, as you've interacted with these washing facilities and begun to become more informed on what happens in that transportation step, what has been most eye-opening for you from a research side, or maybe other things that you think we should explore as an alliance beyond the validation of the sanitation, but are there other things that you see as potential opportunities?
I think, yeah, so I do agree with you. Maybe at the beginning they were not on the table, but I think this study demonstrate that they are willing to be engaged. So I would suggest the Alliance to leverage this relation that we establish in order to continue the conversation. I feel also that they were reassured by the fact that at the end, for what we sample, they were not extremely high log to deal with, but nevertheless, that doesn't mean a lot if we talk about foodborne pathogen, because we know that even one cell of certain pathogen can be can create an illness. Nevertheless, I think they were encouraged on the fact that we were there, we worked with their schedule, and they were open to suggestions. So I feel that if the Alliance worked with them for a common SOP, they will be open to work more.
Absolutely. Yeah, it's nice to see these industry collaborations lead to impactful research, which really, again, is at the core of one of the Alliance's missions, is connecting those in academia to the spaces within this industry where research is needed and is super impactful. So thank you for leading that from your lab. And I know everyone's really excited to see the final results from the study and the next steps following this, I think, really groundbreaking study as it relates to transportation of ingredients utilized in the pet food and rendering industries. If there are other questions, feel free to unmute yourselves or type them in the chat. I also know that, as Dr. Trinetta mentioned, this study is ongoing, so she's looking as well for suggestions, thoughts, comments. And if you don't feel comfortable sharing those in a webinar such as this, Dr. Trinetta, if you wouldn't mind in the chat sharing your email address so that folks have that and can reach out to you directly. And from an Alliance perspective, we do plan on having Dr. Trinetta join us as the study wraps up to share those final results and really think intentionally as an entire Alliance about what next steps might look like to continue this research and to continue to see impacts in this space. Because again, those collaborations that exist are impactful not only to move the needle as it relates to sanitation during transportation, but to identify other opportunities for research with academic partners. So if there are no other questions, I just want to thank you again, Dr. Trinetta, for joining us today and sharing this information. It looks like Namal has a question. So he read my mind. He listened to my cue. Would it be good to evaluate the effects of additives like sodium bisulfate?
And for where would you add those additives? Into the raw material? Or yeah, can you elaborate a little bit your question, please?
And feel free, there you go, as a pH
Yes,
reducing.
pH reducing agent. Yeah, but thank you for the suggestion. I might take notes on that. Some bacteria might be affected by pH, definitely. So you would get probably a higher log reduction. Yeah, thank you.
All right, feel free to continue asking questions or send them directly to Dr. Trinetta in the email address that she's provided. And perhaps most importantly, is I would encourage you all to continue to participate in the working group meetings, the working group conversations, and the collaboration between the Pet Food Alliance, Pet Food Institute, and the Fat and Protein Research Foundation really led to not only the funding of this proposal, but provided Dr. Trinetta with access to facilities as well as industry experts to guide the proposal and make sure that it's most effective and the results are most likely to be implemented once they're completed. So we really rely on industry members participating actively in those working groups, sharing their valuable perspectives and opinions and research needs that they have relative to the space voices they represent and the industries they're coming from. So again, please do join us in November in Auburn as we continue our breakout fall working group meetings, and then we'll convene again in Kansas City in May of next year following the Pet Food Forum with another technical meeting. Hopefully we'll see Dr. Chinetta at that meeting to see more updates about this research, and then we'll talk about some of the other research happening across the Alliance and across the industry. But thank you all again for sharing some time with us this morning or afternoon, depending on where you're at. And we hope that you join us for our next webinar, which will be in November, focusing on some advances in measuring peroxide values. So look forward to seeing you later this fall, and have a great rest of your week.
Thank you.
Thanks.